So, let's start. Last time we talked about scoring matrices and showed you how difficult this problem is. And there is no unique, universal best scoring matrix. Biologically, it's very difficult to understand, come up with a matrix which fits best the real evolution. Today, what we're going to do is, given a scoring matrix, given a certain match score, like Blossom 62 matrix, which contains match and mismatch scores, and gap penalty, how are we going to analyze gaps? Given such a scoring matrix, today we are going to find out how we can find the best alignment, meaning that an alignment between two sequences which matches the amino acids, respective amino acids, which maximizes the final score of the alignment. Okay, so this is, and we are going to talk about dynamic programming solution to that. Dynamic programming, I mean, the name is not, it does not make any sense actually about it was used like a code word, I guess, to specify a certain algorithm. In general, if you haven't seen in algorithms class, dynamic programming is a technique, it's like a divide and conquer technique, which consists of a recursive solution to a problem. And when I say a dynamic programming algorithm, we're not going to write any programs. I'm not going to give a pseudocode of an algorithm. This is, let me tell you, this is the dynamic programming solution to the sequence alignment problem. Okay, the dynamic programming is basically consisting of equations which determine, we define a certain, we first need to define them. The Fij, we define a certain variable named F, okay? And Fij takes two parameters. We also determine, we need to define these variables, F and S, that we are going to use in our dynamic programming solution. And these three equations, as you can see, there are three equations there. Fi0 is this, F0j is this, and Fij is this. Okay? We provide three equations which are called recurrence relations because if you see the definition of Fij, it's composed of also it reuses F. Okay? The F function is reused within the definition of F. Fij is computed by looking at the F values of Fij-1 or I-1j-1 or I-1j. Okay, so it's a recursive definition, and that's it. This actually is a full solution. These equations are a full solution to the sequence alignment problem. It gives you the maximum score of aligning two sequences. So when you are looking at if you look at the algorithm, if you look at a solution to a problem which is provided by using dynamic programming, this is what you get. These are the equations. Now, the question is, if you're new to this algorithms area, if you don't know dynamic programming well, there will be a lot of questions in your mind. Okay, first of all, how does this solve the alignment problem? What is the alignment? What do you get at the end? How is this an algorithm? If you may write these F as separate as a function in your C program or Java program. This F may be defined as a function which takes two parameters, and you can write it as a recursive function, like Fij is equal to. You may call the function itself from the function, but if you do it like it, your program is going to take a lot of time. It may be exponential. Okay, this is not going to be the correct implementation of a dynamic program. One of the most important properties of a dynamic programming solution is that it reuses previously computed values. For example, if in one function call you need F34, okay, this is our function, and somehow in this function call we need F3, 4. In a recursive solution, computes this, makes a call to this function, and computes the resulting value of this expression. And if you need the same value, if you need f34 in another branch of this recursive calls, you may need f34 again. Okay? And if you need it again, a recursive solution, what do? You call the function again with the same values. The same branches that are going to be needed to compute this f34 is going to be taken twice. Okay, if you need it twice, you are going to, so basically in C or in Java, if you make a function call, that function call is executed until you get a value for f34 in both of these f34s. But the most important property of dynamic programming is that it prevents these additional calls. If we have computed this f34, why not save the result of f34 in a table? If we need it again, we may reuse it by looking at the table. Okay, so that's the main efficiency gained in dynamic programming. It's not a recursive solution. That's the difference from other recursive solutions. In recursive solutions, it may take a lot of time. The complexity may be huge due to these additional repetitive calls. In dynamic programming, when we write the function itself, we are not going to be writing the function as a recursive function. At the end, you'll find out that dynamic programming solution is nothing more than just filling a table. Okay, you'll just fill a table starting from the initial cell. We are going to initialize the first column and first row, and by making use of the known information currently in the table, we are going to start filling up the table until all of the table is filled. And for dynamic programming, for sequence alignment, global sequence alignment, the size of the table is going to be n times n if one of the sequences is of length n and the other sequence is of length m. We are going to be filling an n by m table. And to fill in each entry on this table, we'll need constant amount of operations to find what the value of a certain cell is going to be. We are going to look at 3 choices each time. So filling each cell in this table is going to take 1 time, meaning that regardless of your sequence length, let's call 1, okay? And regardless of your sequence length, to fill in a single cell of this table, we are going to need constant amount of operations. You are going to do 3 lookups. You are going to compare 3 numbers and choose the maximum of these 3 numbers, as indicated by this max function. And actually, if you look at these recursion in detail, the first 2 equations, fi0 and f0j, are the equations which are not recursive. It uses a certain lookup to an S table. And actually this SAB is going to be an entry in the Blossom 62 matrix. Okay? S is actually the Blossom 62 matrix. The scoring matrix is itself. It basically says that what is the score of aligning amino acid A with amino acid B. So this S is a single, it's a lookup table. If this is your S table. Let me do that. So this is what S is. For example, we will be asking what is the score of SWA. Okay? So SWA, to find that out, we're just going to go look at this table. AW is -3. Okay? So that's -3. So in this recurrence relation, I'm going to get back to all these. Okay, just now jumping ahead actually, just to show you how the flavor of dynamic programming. So the first 2 equations are used to initialize the first row and the first column of this table, because they say that fi0. So if I is the row index, okay, this means that the 0th column meaning that this is the first column. This is the initialization of the first column, and this is the initialization of the first row, the 0th row, okay, in our table. The indexes start from 0 in our table, so initialization of the first column and first row can be just done by looking at this blossom matrix, blossom 62 matrix, the scoring matrix. We're just going to look. Actually, we will not even need to look at that, because this, look at this, what this S contains. What does this S say? It says that what is the score of aligning, so this, the ith row in the first column. So that's if you analyze this first equation, let's try to understand what it's saying. So we are filling the table of size n by n. So this may be our n, this may be our n, okay? And n rows, n columns. Actually, let's see. From I is equal to 0 to I, and here, so there are some missing things here. Actually not missing. The sequences are denoted by A and B. Capital A denotes the first sequence, capital B denotes the second sequence, and both of these sequences, the first one contains I characters from 1 to I, so it's actually we're going to have an I times J table according to the notation on this slide. So we have an I rows and J columns. And if you look at this equation, actually the sequences start from index 1, okay, from 1 to I, but we also have a 0th column and 0th row. Means that actually our table contains I plus 1 rows and J plus 1 columns. Because for the initial 0th row and 0th column, they do not correspond to a certain character position in these sequences. Okay, so this is what we have. We initialize this table. We have a table. Okay, the first column and the first row they are indexed with this 0. Now, if you look at the first equation, fi0 is equal to a summation term. It says that. So first, we also need something actually that is equal to 0. Okay, for k is equal to 0, a0 is undefined. There is no such thing. Actually, we should probably some certain mathematical slight incorrect parts in this equation. Maybe we can have an additional f00 is equal to 0. Okay, we need to first say that this part is 0. Okay, the first top left cell is going to be initialized at 0, and then the rest is going to be the rest of this column is going to be defined by the first equation. So f10. So what is f10? To find f10, what we need to do is, so here f10 is equal to summation of, so the index of the summation term is from 0 to 1. Okay, actually we can correct from 1. So the correction I'm going to make to these equations is this. This index of this summation term, which is k, I'm not going to start it from 0. I'm going to start them from 1. Okay, so my correction that I'm making to these to the slide is that k changes from 1 to I. In both here k changes from 1 to j. And the main reason for that is when k is 0, a0, there is no such thing. Okay, what's the 0th character of the first sequence? Because our sequences, the indexes start from 1. Okay, so a0 and b0 are undefined. That's why to be mathematically correct, I start the indexes, so from k is equal to 1 to I, okay, the first equation, s ak, comma, a gap. Okay, this is what the first equation is. Actually, if you have written these indexes correctly, you don't need the second part of 0 from k to I. So this is the first equation. F10 is equal to k. Sorry, here it's going to be from 1 to 1. Okay, if Fi0 is this. Okay, Fi0 is equal to from k is equal to 1 to I, and the summation of these scores. And what is this score? Actually, this score does not exist in the Blossom matrix, because this score asks for aligning an amino acid with a gap. Okay? So, what is the score of aligning a single letter in an amino acid with a gap? And the Blossom matrix only contains the alignment between amino acids. It doesn't have the gap. So, but what is this? You know, this is the gap penalty, right? So this is our gap penalty, aligning an amino acid with a gap. This S. The result of this S is the gap penalty. And the solution that we showed on this slide actually uses a linear gap penalty, meaning that opening and extension penalties are the same. And this summation, since we are using a linear gap penalty, okay, if we use a linear gap penalty, it's also written here, we assume linear gap penalty. This is the solution for linear gap penalty. So we have a fixed gap penalty like this. Gap penalty, for example, -5. Okay, because we have a single gap penalty, constant gap penalty. It's linear, opening and extension are the same, all -1. Now, the score of aligning any amino acid with a gap is always going to be -5. Okay, whatever that amino acid is, this gap penalty does not change from an amino acid type to amino acid type. Okay, if you align A with a gap, it is -5. If you align W with a gap, it's again -5. So actually, this summation can be just simplified as I times -5. Okay? So how many letters, whatever it is, because we are summing each time from 1 to I, there are I such terms, and all of those terms are -5. So it's equal to I times -5. This is your the simplified version of your column initialization. So it means, it basically says that the first column is going to be initialized to this. -15, -20, and goes like this. I times -5 at the end. So I use this 5 for this index and the length of the sequences. Actually, that's again something wrong here. Again, another thing wrong on this slide is that I is used both as a general variable here to indicate all the rows. Okay, from I from it should be actually what we should have here is that this should be this just initialize it. It doesn't initialize the whole column if you use I like this. It just says that what is the last entry in this table is this. Okay, if you use the same I variable that you use to indicate the length of the sequences, Fi0 corresponds to this cell, right? This is Fi0. But the purpose of this equation is actually to initialize the whole column, so we should use a different variable name. So we have identified a couple of errors on this slide. I mean, these seem to you like maybe details, but if you're writing things formally, you need to make sure you don't have these errors. Okay, I mean, because for not to have any ambiguities, if you want to, you need to be mathematically fully 100% correct, so that your equation is on what your algorithm is understood correctly. So this is we should have used something or maybe you can change these from 1 to n. Oh, actually, it's not wrong. You should have corrected me. So there's nothing wrong on this slide about INJ because this is how we define Fij? Sorry about this. Okay, I should have. So actually, the sequences themselves may be of length m and n. Okay, so this part can be n. Okay, n plus 1, n plus 1. So as this definition, this Fij just defines an arbitrary cell which is indexed by ij in this table. Okay, so Fij was a general definition. Ij, those indices can be used to describe cell I, row I, column j, so an arbitrary cell. So I and j are indices used as these variables. But what was missing from this slide was what is the maximum length of these sequences? What is our table size? What is the length of sequence A? What is the length of sequence B? This is missing. Not an error, but it's missing from this slide. So we can go back and use m plus 1 and m as our sequence length. So this says that the ith row in the first column basically is just can be initialized with this regular gap. So if you think that what this means, okay, if you think about this, it means that the first column, for example, I'm going to come to that later. Okay, the alignment, I haven't talked about how we represent an alignment yet, but the definitions are here really important. So Fij, the optimal similarity of A1i and B1j means that we define the first prefix, the prefixes of A and B up to ith and jth characters if we know how to align them optimally. Okay, this means that. So this Fij, this table cell, Fij, denotes the score of the optimal alignment of this sequence with this sequence, not having yet considered the rest of the sequences. If we can, what is the optimal way of aligning the first part of the sequences? Okay, Fij corresponds to that. And you are going to see by using these equations, we can initialize the column and the row, and then the Fij, the second general equation, will help us to fill in this table. By using these three of those, we will be able to fill in this. And by using these three, we'll be able to fill this part. So going one by one like this, we'll be able to fill the whole table. And at the end, the entry here, which is equal to Fn, m, okay, the entry in the rightmost table cell here is going to denote the optimal way of aligning the complete sequences. So it's like we divided the problem, it's like a divided computer approach to find the optimal way of aligning the complete sequences, which are like n and m. We make use of some smaller sub-problems. For example, what is the, if you know the optimal way of aligning a smaller sequence, the first prefixes of the prefixes of these sequences, you can make use of this information to grow the alignment one letter by one letter, and at the end you'll be able to find the value of this table cell, which is going to give you the value of the optimal alignment, okay, the score of the optimal alignment. So we are going to come to that again. Okay, just the main reason I showed you this slide first is that dynamic programming is nothing but these recurrence relations. Okay, so if you're going to propose a dynamic programming solution to another problem, all you need to give to the reader is this set of equations. What is your recurrence relation? And this equation defines everything. It's a solution to the sequence alignment problem. Now let's come to the table idea. Let's first introduce this table idea. Before dynamic programming, this. Okay? So on a table, we put one of the sequences in this top row, and the other sequence, so these are two different sequences, therefore we do not necessarily have a rectangular table. We may have a very short sequence, and we may compare it with a very long sequence. So we may have not necessarily a square table, it may be a rectangular table. And these are two different sequences. And within these, so we have a table, and we may put a dot whenever we have a matching amino acid pair for this corresponding cell. For example, look at this. We have it here. I have a G. This G could be matched to this second G. So I'm looking at this row, this G in this at the rows, and this G can be matched to the second G in this sequence, the fourth G in this sequence, and it can be matched to all these G's that exist in this sequence. So what we do is, in this row, we put dots corresponding to these matches, and the unmatched, mismatched parts are left empty. And at the end, what we are going to get is a matrix filled with dots, maybe lots of dots. Now, the sequence alignment problem can be considered like this. Provide a sequence of diagonal matches, which means that, for example, if you look at this region, okay, imagine this short stretch was all marked with these dots. What it means is that this C, for example, can be matched to this C, and this G can be matched to this G. So there's a consecutive sequence of matches. This diagonal represents that. Okay, there's a consecutive, there's a sequence of matches, and this match run, we can also call it a matching run, a run of matches, the consecutive matches along the sequences. And this diagonal will represent that. And what about a horizontal line? What does it represent? A horizontal line represents this. It means that you have matched, just look at the leftmost diagonal part of this horizontal line. So for example, an A is matched with another A. Just before this horizontal line starts, we have matched an A with an A, and then for this one, we stayed at this column. Okay, sorry, we stayed at this row. We do not go to lower rows. We are still, horizontal line means that you are still, the last thing that you matched was that A. Okay, you do not progress in this sequence that is here. You are still at that A, but you try to match all these C, 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 T with something. So if I didn't progress on the first sequence, what is this ACC matched against? Gaps, exactly. So it means that a horizontal line means putting a gap against these letters at the top sequence. It's like while you are moving, so an alignment will be a trail, okay? And this starting from the leftmost cell to the left top cell to the rightmost bottom cell, we are going to find a trail, a path. And this path is actually the alignment itself. Whenever we have a diagonal, if we go, when we follow a diagonal route, it means we are matching corresponding amino acids. When we follow a horizontal path like this, it means we are matching gaps against, because we are consuming, when we are going through this path, we are consuming letters from both sequences. If you are going a diagonal path, you are consuming amino acids from both of them. But if you are going on a horizontal path, you are consuming letters from the top, but you are still staying at the same amino acid from this column sequence. It means that you are putting gaps again, or you are matching these amino acids with gaps. And similarly, we could have, examples are really short here, but we can have vertical paths as well. And this vertical means that we are still at the same amino acid at the top sequence, and we are consuming some amino acids at this one. We are matching these amino acids against gaps. So we will be basically go along the path, and that path may look like this. Okay? So we may have horizontal, so we may have, so the path may be like this. So this is amino acids are matched with each other. This is gap against these. So for example, I have AAA. So gap against these AAAs. Let me just try to give an example. Yeah, GGG. Okay, so imagine these two sequences, G, 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 A, C, 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 and T, T, T. This is our first sequence. And the other sequence has G, 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 C, 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 A, and T, 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 T. Okay? So it seems like this first, the middle AAs and CCCs are swapped. So here, in this alignment, what it corresponds to, we match the first G's, is shown as a diagonal path here. And this path here, okay, says that we are still here in this sequence, and we proceed on the second sequence. So if you write it as an alignment, it's going to look like this, the final alignment. So G's are matched with G's. Then I have AAAs, I have gaps against them, okay? And then I'm here, in both sequences I'm at the beginning of the C's, I can match these CCCs, and then I'm right here in these sequences, okay? So I'm going to have the other sequence matched against with gaps, and at the end I'm going to have the TTTs matched. Okay, so a path in this table represents such an alignment, and we may have gaps in both sides. And if this is left as an exercise, how many possible different paths can you have in such a table of size n by n? Okay, how many different paths can you draw? And it turns out that it's much larger than it's exponential. So you can have an exponential number of different paths on this table. So means that there are many, in the solution space, there are many different alignments. In the solution space, many alignments exist. Okay, so just try to work out how many possible paths you can draw on such a table. What makes this complicated is the presence of gaps. You can have any size gaps, you can have them anywhere. Okay, so it's, in other words, if you at the end you'll come up with a conclusion like this, and any subset, any subsequence of this can be matched with any other subsequence here. So that all these possible subsets, I mean, the possible subsets of a larger set is the number of these sets are exponential, and that's probably the reason for this exponential number of paths. Now, how are we going to find our solution is able to find this in polynomial time? You only do it in n times n times. In n squared time, we'll be able to do this. Now, here is an observation. This is the additivity property of score, which will help us to divide the problem into subproblems. So the score of aligning two complete sequences like this are additive, meaning that if we know For example, if you know that the first I characters of the first sequence has been aligned with the first J character of the first sequence, the second sequence, and the rest of the sequences are aligned separately. Okay, if somebody gives you two separate alignments which divides these sequences in a certain location, the sequences are chopped into two pieces, and we are given the respective alignments separately. The total alignment, if you just combine these two alignments together, because our scores are local. If you look at our scoring matrix, what we have is match score, mismatch score, and gap penalties. And these penalties, especially in the, just assume linear gap penalty right now. Fine gap penalty is more complicated. But if you just assume linear gap penalties and these mismatch scores, what you'll notice is that this score is the summation of all these individual scores. Okay? Whether you have a gap at that location, whether you have a match at that location, you just sum up, you add these scores to come up with the final score. What this means is that the score here does not depend on the score somewhere else. It's always -5. This -5 after you make the alignment. After you make the alignment, this -5 here does not change based on your decisions elsewhere. These scores are computed in a locally for just that sequence. Means that if somebody asks you the score of the whole alignment, it's just the summation of this score, score S1 and score S2. So our scores are additive because they're computed independently, locally at each region. So the score of aligning the complete sequences is actually equal to the score of aligning the prefixes up to I and J, and the suffixes up to starting from I plus 1 and J plus 1. Okay, so the score is alignment is additive. And there are two types of alignment generally. We are going to consider the first part, the global alignment first. The global alignment is the problem of aligning the whole sequences without, you need to match every amino acid of each sequence to either another amino acid or to a letter. You are not allowed to discard some of the part of the sequence. All the sequence letters, amino acids need to be aligned in global alignment. And this kind of alignment is usually used for strings of sequences of similar size. And local alignment seems like a much more complicated problem, but we're going to see the solution is really similar. Finding similar regions, and if we consider the sequences completely, the overall sequences may not be very well matched. But there may be certain local regions. I mean, discarding some of the large portions of both sequences, you may find a small region which has a high matching score. But due to dissimilarities in other regions, the overall alignment, if you do global alignment, our score may be very bad. Because most of the regions of these sequences may not match very well. However, if we consider a local portion, a subsequence in both of these sequences, we may get a match. So global alignment oversees such small matches. It cannot capture these small matches because its goal is to align the whole sequences, to optimize the score considering the complete sequences. But it's biologically, a more biologically motivated problem is the local alignment problem, because sometimes we may have sequences which are similar. They may share a single protein domain, but the other domains may be different. The sequences may be completely different. We may sometimes need to find that shared domain which has a similar sequence. So we may need local alignment, which shows similar regions only subsequences of these two sequences. So here, the most important property of dynamic programming solution is that we have limited choices. If somebody gives you the optimal alignment up to a certain point, so imagine we have two sequences, and imagine somebody gave you that, somebody told you that the best way of aligning the first I characters of this sequence to the first J characters of the sequence is given to you. Okay? Somebody aligned this part for you optimally. You know they aligned. Now, the question is, if you want to grow this alignment, one more amino acid, what are our choices? How will the alignment look like if we want to find out what is the optimal way of aligning, for example, I plus 1, J plus 1? Okay, what is the optimal way of doing this? So we see that even actually looking at this intermediate space, even if you just look at the first position of the alignment without having aligned anything, you didn't align anything yet, but somebody asks you, what is the first position of the alignment going to be? Okay, I mean, you do not know anything about the complete sequences. But given two sequences like CA, CGA, and CGA, how can, in the final alignment, in the optimal alignment, what is the first position going to be? And you realize that there are three possibilities. I mean, these alignments do not, you need to obey certain rules, and those rules is that an amino acid sequence needs to be aligned against another amino acid, another nucleotides in this example, or it needs to be aligned against a gap. So therefore we have these three positions, three options. The first position alignment of the alignment can be either CC, gap C, or C gap. There cannot be anything else. Okay? I mean, if you look at the final alignment, the first position needs to be one of these. But right now, by looking at the overall alignment, we cannot tell for sure which of these options are going to be existing in the final alignment. Okay? Because the thing is, I'm not saying that at this point we have these three options, but we cannot tell right now which one is going to appear in the final alignment. We cannot tell it right now. But it's going to be one of these three. But we can score each of these first positions. Okay? Each of these first positions, if our gap penalty is -1, and if our max score is 1, this first position, such an alignment, aligning the first two letters, is going to score 1, and the remaining sequences will be this, and aligning gap with this C is going to be -1, and the remaining sequences will be this. Okay? Now, here is how we can actually find what the final alignment, optimal alignment contain. Out of these three choices, which one our alignment is going to contain. How can we do that? If somebody gives you the best way of aligning the smaller sequences, okay, if we have the optimal score of aligning ACGA with GA, okay, imagine the optimal way of aligning ACGA with GA is 0. Okay, I'm just assuming GA is aligned with GA, so I have two gaps, -1, and two matches, 1, 1. So imagine somebody, we have run the algorithm and we have found that the thing you should realize is that we are solving a smaller problem here, because these sequences are one letter less. Okay, the other is a larger problem. So we reduce the problem to a smaller problem by assuming C is aligned with a C. So the overall alignment, okay, if we have taken this first choice, the overall score of the alignment would be 1 plus 0, right? This 0 is the alignment of, is the score of aligning ACGA with GA. And if we know that, okay, if we know this right part, which is 0, the overall value of the alignment would be 1. Everybody agrees with that? This comes from the additive property. If we have taken the second choice, what is the score of GA with CA, CGA. If again we see that we can do it manually by hand easily, I think the best score we are going to get is by aligning these two GA parts. So +2 from that and -3 from these 3 gaps. So the aligning, best alignment of these two subsequences would be -1. So taking this path, taking this option as our first position, our score would be -1 plus -1, which is -2. Now we can actually compare two global alignments. Taking the first path, taking the second path to the end. The first path is better. So aligning C with a C is better than aligning C with a gap. And actually, to be able to find the actual optimal answer, we need to also do this. And this one, if you look at that, so CGA aligned with A, probably we're going to get a score of 2 from here. Okay? 3 matches, 1 gap, 3. This is going to be -1. 3 minus 1 equals 2. So actually we have a better alignment. So as you have seen, while knowing the solutions to the alignments, which are smaller sequences, we were able to compare our first position choices, and at the end it turns out that aligning the first C, this with a gap, we can obtain a score of 2. Which is comparing these 3 numbers, sorry, this is -2. So 2, -2, and 1. 2 is the maximum of these, so the optimal way of alignment is going to have to choose this path, which is going to align with this C with a gap. Okay, so the dynamic programming algorithm is based on this idea. We're going to decrease the problem to a smaller size by making use of this fact. I mean, we do not have much choices. You either do a match, you either do a gap, either in this sequence or the other sequence. You do not have any other option. So we are going to fill a table by making use of this fact. We are going to continue tomorrow about